Okay, so let me get started on mass spectrometry for biomedical research, and I will show two examples. One of them being published in the special issue in bioengineering, and that's on prostate-specific antigen. So that's going to be the first part of my talk, revealing a novel glycosylation motif on this protein, and then the second part will touch on antibodies. So this is very much in line with what Gordon just presented, looking at antibodies in COVID-19 infection. Good. And let's start on prostate-specific antigen. So this is a marker in prostate cancer. And it's used for, let's see, I'm still struggling a bit with the technology. So it's used, the PSA test measuring the concentration of prostate-specific antigen is used for detecting prostate cancer. Now, there's ample indications that also the glycosylation of PSA may be relevant for detecting prostate cancer early on. And we were looking into the glycosylation motifs of this protein. And to do so, we generated glycopeptides, as indicated here in an example. We measure them by CEMS, so by capillary electrophoresis. We get a wealth of glycopeptides. And what I'm showing here is some glycopeptides indicated with this red trace, where we had a strange composition, which we could not match with a specific glycan structure. So you see the question marks here. We were doubting what are now exactly the glycan structures we have here on prostate-specific antigen. And to do so, we chose not to analyze only glycopeptides, but we really combined an array of different methods. So here what we did is we released the glycans from PSA, from this protein, and we did tandem mass spectrometry, in this case in negative ion mode, after porous gravitatized carbon separation. And I just want to highlight two signals which we observed here, the one indicated with the green arrow over here. So this is a loss of 250 Dalton from the entire glycan unit, indicating there's a terminal monosaccharide with this specific mass. And we found also a fragment ion here at 249, confirming the presence of this terminal motif. So we could rule out conventional sialic acids because they have a different mass of 291. So what is this 250 modifications? It's cathodeoxynononic acid, so KDN, let's say. And it's kind of a deamidated, non-acetylated version of conventional N-acetylneurominic acid. So we were quite excited about this finding because this sugar had not been reported on human proteins before. So it's really a novel finding we published here in engineering. And here is the structural difference. So we don't have this N-acetyl group present in the case of KDN, but have a hydroxyl group. And just to give some more flavor of what we found here, so we found a whole group of glycans, all shown here, some of them having only this kind of modified sialic acid, the KDN on, but others having kind of a mixture, meaning on one antenna they have the conventional sialic acid, and on the other one they have this novel found KDN motif. And we found them with a co-fucose, but also in absence of a co-fucose. We found isomers for each of these compositions, so a whole family of these KDN glycans. And we confirmed that by tandem mass spectrometry, as indicated here. So we have this mass or ion at 657, indicating the conventional antenna with an acetylneurominic acid, so the conventional sialic acid. And then we have this novel one at 616. So pretty clear indications for this novel modification. And a key aspect for elucidating these modifications is the combination of different mass spec tools. So we measured glycopeptides by capillary electrophoresis with mass spec, by reverse phase LCMS with a mass spec on two different platforms. We did this with linkage specific modification and without, and we also did release glycan analysis, as I showed on with this PGC porous gravitatized carbon coupling. And overall, this allowed us to unambiguously confirm that we have this KDN modification on N-glycans on prostate-specific antigen. So this brings me to the summary of the first part of my talk, and that is actually that we demonstrate the presence of KDN of a norphocylic acid on the end glycans of prostate-specific antigen. We found a whole family of these end glycans with one or two sialic acids, with and without corphucose, and present in different isomers, so really a wealth of different structures. Now, in the meanwhile, we looked at different glycoproteins because we obviously wondered, is this now a unique modification of PSA, or is it found on other proteins? And yes, we found it on a few other glycoproteins, but it doesn't seem to be a very common modification on human glycoproteins. Now, of course, the question is, what could be the function of such a modification? And what we think is that such a KDN may be less amenable to sialidases. So I think this could easily decrease the desilylation of proteins, and in that way increase their stability, because desilylation is often the first step in removal of proteins from the circulation. Also, it has been demonstrated that humans have antibodies against KDN motifs. And so we think that the KDN on some of the proteins may result in antigenicity, so binding by antibodies, maybe clearance or inflammatory responses to these KDN glycans on cells or on proteins. Now, as I indicated, we are looking into the expression of KDN in different human tissues, but also on different proteins. And of course, we're wondering whether KDN, also with these inflammatory responses, may be involved in cancer processes or other malignancies. So this was the first part, summarizing this engineering paper by Wang Wei, so a PhD student in my group. And now I want to take the next couple of minutes to look again at antibody glycosylation, which already was addressed by Gordon. So here we have the structure of an antibody within the Fc portion, the glycans, and Gordon already showed that these glycans reflect aging, reflect inflammation, and there's a wealth of these different structures, and you then can distill certain glycosylation motifs. Now, what we did during the COVID pandemic is we looked at these antibodies and their glycosylation using a modification of an ELISA assay. So what we do is we put serum or plasma of infected of COVID-19 patients, let's say, on a 96 well plate, which is covered with the S protein of the virus, recombinantly produced. We then elute the antibodies, digest them. We measure these glycopeptides, and we register all the glycosylation features. And what we noticed is that we have, looking at these anti-S antibodies, IgG1 antibodies, we see that they have a lack of corphucose, so that's kind of these glycan here, and this glycan, all of them lacking the red triangle, while the total or bulk IgG has this corphucose present. And this is kind of done here for close to 200 patients. So the total IgG has high levels of fucosylation, while the specific IgGs have either a high or a low level of fucosylation, so a vast spread, and this may reflect different functionalities of these antibodies. And why different functionalities? So this has been known for close to two decades already. So what we know is that antibodies lacking the corphucose are very potent inducers of cytotoxicity. That's shown here. An in vitro assay with an antibody, the antibody lacking the corphucose induces killing of cancer cells with a corphucose. It's a lousy inducer of cancer cell killing. And what is now the structural background for this? This has also been published quite some years ago already. So we know that without this corphucose present, so in this case, we have a close engagement of the antibody and of the 3A receptor on immune cells, so lots of hydrogen bonding, high affinity, and this results in the recruitment of immune cells and in the killing of cancer cells. What does now the fucose do? It blocks these interactions less hydrogen bonding and lower affinity, so then these interactions will not form. Now, in the case of COVID-19 infections, we saw that indeed these antibodies with a low fucose, if you form immune complexes with these antibodies, those are the best inducers of interleukin-6 in an in vitro immune cell activation assay. So indeed, these antibodies with a low fucose induce inflammation in a COVID-19 setting, and we think this happens in the lung. We then looked longitudinally following patients over days and weeks, and we saw that this low fucose phenotype is actually only occurring in the first couple of weeks, and then it's vanishing. So this is an interesting aspect, showing us that antibody glycosylation is not a static thing, although they have a half-life of 3-4 weeks, these antibodies, but at the start of an immune reaction, we have a very dynamic process, and glycosylation, not only fucosylation, but also characterization, silylation, bisection. So overall, the glycosylation of this antibody is changing very rapidly. Now, just to summarize also the second part of my talk, I showed you now data for COVID-19, which is specifically for the S protein, which is membrane embedded. And actually, we looked at various immune settings, and we found a pattern that membrane-embedded antigens often induce these low fucose responses, whilst antigens which are soluble never appear to induce low fucose responses. But in this case, where you have a membrane-attached antigen, you indeed often have these low fucose responses, and we think that this is actually a double-edged sword. And in some cases, such a low fucose response with its potent induction of or recruitment of immune cells could clear an infection very potently and rapidly, but in other cases, it may induce excessive inflammation, as in the case of the lung of COVID-19 patients. So indeed, it's a potent trigger or switch in the immune response, which can have very different outcomes and can be beneficial, but also lead to adverse outcomes. So this was actually what I want to tell you. And the first part of my presentation was indeed the work by Wang Wei from my group, PhD student. So she discovered this novel modification, KDM, under guidance of Guinevere Lachwein Kammeyer from my group. And this is the whole team at our department. And this is what I wanted to share with you. Thanks for your attention.
工程创造人类美好未来。